Assalamu alaikum wa rahmatullahi wa barakatuh. Good afternoon. It's my pleasure to be with you today to give this talk about intermittent fasting as a driving force for anti-tumor immunity. Indeed, I really wanted to be with you, but for unforeseen issue, I couldn't come and make it to Sharjah University. And I'm following the meeting. It's a very successful one, and hope everyone enjoy it. With you, Mohamed L. Salem, Professor of Immunology, Faculty of Science, Tanta University, and Director of Center of Excellence in Cancer Research, and the Chairman of Egyptian Society for Cancer Research. I founded the Center of Excellence in Cancer Research in 2013, and from that time I focused on cancer immunotherapy, plus of course other topics on anti-cancer drug discovery in cancer, immunotherapy. I focus more on dendritic cell-based vaccination and the T-cell-based therapy, and explore how cancer cells escape from anti-tumor immunity, focusing on myeloid-derived suppressor cells and regulatory T cells. We use both animal models and also human biological samples from cancer patients. The outlines of my talk today is about fasting and anti-tumor responses, fasting and anti-tumor immunity, fasting and immunosuppressor cells, fasting and stem cells, and then future direction. Due to the limited time, I will just give overview of each topic of these. The effect of different periods of intermittent fasting it's shown here in the case of short-term IF and long-term IF. In case of short-term IF, it induces ketones, autophagy, DNA repair, upregulated certain microRNA, and increased glucose protein synthesis, mTOR expression, and remodeled cell growth and functional issues. Of course, this depends on the periods of caloric intake restriction and the period of recovery after fasting. So eating, sleeping, and then fasting. The longer-term IF is similar effects, but with increased insulin sensitivity and body resistance, improved blood glucose homeostasis and lipid metabolism, and reduced abdominal fat and inflammation. This is the general overview of the effects of IF, either short-term or long-term. I'm not going to repeat. Of course, everybody talked about the effect of fasting on the body and the different forms of fasting. But surveying the studies I worked on, I can say fasting I can divide it on three main categories, which is fasting on alternate days, which is short-term intermittent fasting from 8 to 16 hours of daily fasting, then prolonged intermittent fasting, entire day fasting, or fasting within a time frame, for example, 1-2 days per week of total fasting, meeting up to 25% of calorie needs. So this is the different forms of IF. And when I went over the progression or the timeline of or the history of IF research, I found it started more than a century ago, and most of these studies actually focused on two things. One is I'm focusing on anti-tumor response and immunity. I found it's mostly in experimental animal model. For example, more than 100 years ago, they first described the beneficial effect of fasting on tumor in animals, and then in 1997, caloric restriction has a significant effect on aging, and so on. Then the studies started to look at immune response and the mechanism behind the anti-tumor immunity and the anti-tumor response. Until recently, several studies looked at the impact of fasting on regenerative properties of hematopoietic stem cells, and also tissue-specific stem cells, especially in GIT, as well as to look at how fasting affected the immunosuppressive microenvironment of tumor microenvironment. So it's a very long history, started 100 years ago, and is still working very fast and with accumulating very nice and interesting data. The hallmark, as you know, the hallmark for cancer, which is about from 10 to 13, starting from evading gross, sustaining proliferative signaling, evading growth receptors, avoiding immune destruction. All these hallmarks, cancer hallmark, can be interfered with fasting, especially certain hallmark like avoiding immune destruction and cell proliferation, and the tumor promoting inflammation. Fasting, the effect of fasting on healthy cells is a little bit different than the effect on cancer cells. In case of healthy cells, fasting release differential stress resistance. However, in cancer cells, it enhances differential stress sensitization, making cancer cells to be more susceptible to chemotherapy by increasing DNA damage and oxidative stress, especially either chemotherapy or radiotherapy. That's why short-term fasting can enhance anti-tumor immunity by interfering with most of these cancer hallmark. Overall, the effects of prolonged fasting on different system of the body in cancer patient include liver, brain, adipose tissues, and the muscles. In liver, glycogen, the increasing glycolysis induce pyruvate and then ketones, and this are transferred into brain and muscles, glucose and the ketones into brain. This induce synaptic plasticity, improved cognition, improved neurogenesis, reduce the inflammation, enhanced autophagy. Ketones are transferred to muscles and also can induce some certain enhanced autophagy and increased insulin sensitivity. This is combined with fatty acids coming from the metabolism of triglyceride in adipose tissue, and this also accompanied with a protein metabolism into amino acids, which then induce gluconeogenesis in liver. So this cycle actually is beneficial for the cancer patient in terms of the signature of metabolic molecules upon fasting. In cancer cells, the fasting induces certain pathways, including MAP kinase pathway, insulin and insulin growth factor. It reduces insulin growth factor, and reduce glucose, and as well as MAP kinase and heme oxygenase. These pathways, certain pathways, induce autophagy in cancer cells. And with chemotherapy induces DNA damage and p53 activation. So the combination of fasting and chemotherapy induce cell death, and this block the immunosuppressive cells like Treg cells and M2 type macrophage, which is similar to MDSC in human, and of course this release the regulatory or suppressor effects on cytotoxic CD8 T cells. So combination of fasting and chemotherapy in cancer patients or tumor-bearing animals showed the beneficial effect by inducing cancer cells, autophagy, and blocking the regulatory mechanism by immune cells. And this is the show the how a combination of fasting and chemotherapy, or even fasting mimicking diet, if MD can enhance the tumor eradication as chemotherapy does alone, but also restore the healthy cells. So this is because chemotherapy, of course, impact non-cancer cells. So fasting and the chemotherapy can induce both tumor eradication as well as restoration of normal cell function and integrity. And this restoration of the anti-tumor efficacy and the restoration of health cells function and integrity has been shown to be mediated by adipocytes under the effect of intermittent fasting. And mechanism is not very clear, but induce certain mechanism like mTOR, AKT, MAP kinase, and other molecules that mediate the both the anti-tumor effect as well as the restoration of healthy cell, normal cell integrity affected by chemotherapy. And this is the beauty of the combination of fasting and chemotherapy. In this table, it shows the overall overview of preclinical studies in vivo reporting immunomodulatory effect of short-term fasting. And this table shows the also studies reported in clinical trials, and most of these preclinical and the clinical studies significantly showed that combination of both short-term fasting and chemotherapy is much better than fasting or chemotherapy alone. Now, how fasting and affect immune response? I can say it's simply by enhancing the enhancers, I mean the molecules and cells responsible for or mediating immune stimulatory effect, and suppress the suppressors, which are induced by tumor microenvironment. In terms of immune cells, the innate and the adaptive immune cells, each type of these cells mediate a certain or play a certain role in anti-tumor immunity. Especially dendritic cells, which bridge, and macrophage, which bridge the innate immunity and adaptive immune cells. And the most important cells here are T cells, and especially CD8 T cells, with the help of CD4, because as you know, CD8 T cells are the killer cells with specific anti-tumor immunity. Of course, other cells like macrophage, dendritic cells, and the natural killer cells, neutrophil, as innate immune cells, play very important roles, but their role are stimulatory in normal situation to enhance the stimulatory effect of T cells. However, this is not the case as I will show you in under cancer persistence. Especially the immune cells are very elastic and plastic in terms of activation upon activation with certain antigen, and of course it depends on the antigen. It can be induced to or primed for what is called Th0 cells, and these cells with certain phenotype like CXCR5 then can be induced into Th1, Th2, Th17, TFH, and each type of these cells a certain function. For example, Th1 is very important for anti-tumor immunity and antiviral, antibacterial. Th2 for parasites, Th17 induce inflammation, autoimmunity, and the TFH induce help for B cells and certain types of T cells. So this elasticity of T cells from naive to effector shape the anti-tumor immunity. And of course, anti-tumor immunity is shaped by tumor microenvironment. So in case if we have apoptotic tumor cells which release antigen, these antigen are dendritic cells uptake this antigen and then processed and migrate from the site of tumor into the nearest lymph nodes to present this antigen to helper T cells and cytotoxic T cells at the same time using certain molecules, MHC class 1 costimulatory molecules. This is normally supposed to happen. However, because tumor cells are self cells, so they don't encode for danger signals. So when we provide danger signals during this process, we can get functional helper T cells and cytotoxic T cells, and the migration, of course, of dendritic cells to mature T cells in lymph node. We have worked on this, and we provided certain danger signals like poly-IC double-stranded RNA, CpG, ODN, certain danger signals which are microbial products, and we were able to get this interaction between dendritic cells and the T cells, and which resulted in anti-tumor immunity after provision of danger signals. However, without danger signals, without any acute inflammatory cytokines, the tumor harbor or has what is called the tumor macrophage. Tumor microenvironment induce certain metabolic pathways, which results in certain cells, accumulation of certain cells I'm going to talk about later on. But overall, the microenvironment, the metabolic signature in tumor microenvironment, including a decrease on glucose, glutamine, arginine, and the increasing lactate and the Kg and increasing acidity, hypoxia, results in inactivation of effector T cells, naive T cells, results in apoptosis, dendritic cells, neutrophil. All cells, neutrophils of course are induced into type 2 neutrophil, and so macrophage. So the tumor microenvironment, all stimulatory cells are induced into regulatory cells, either adaptive immune cells or innate immune cells, and this results in, of course, significant decrease in antigen presentation and interferon gamma, and increasing in inhibitory molecules, and this, of course, block the anti-tumor immunity. So the tumor microenvironment is very complicated, and especially with and rich in immunosuppressive cells and molecules. And this is in normal condition. This is tumor cells and how they induce this immunosuppressive cells from stimulatory cells. However, after intermittent fasting, this is blocked by fasting through IGF-1, IGFP protein binding 1, and all this cellular differentiation from stimulatory into immunosuppressive cells under the effect of tumor cells are blocked and induce anti-tumor immune response. So the situation in tumor microenvironment turned under the effect of fasting, it turns from tumor immune escape into anti-tumor immune response. How this happens, how the mechanism behind that, we can classify it into two effects: decreasing immunosuppression and increasing anti-tumor immunity. Fasting, short-term fasting, decrease CD73 on tumor cells and suppressor cells, also decrease HO, decrease glycolysis, and of course this results in decreasing M2 macrophage, enhancing or blocking the suppressor effect of regulatory T cells on cytotoxic CD8 T cells, and block the growth factors responsible for the release of type 2 macrophage, which is called the MDCC. And so these three mechanisms results in decreasing immunosuppression induced by tumor cells. On the other hand, short-term fasting enhance anti-tumor immunity through regeneration of T cells from bone marrow into peripheral pool and increasing memory T cells, effector T cells, naive T cells, and of course this results by the end of the day into enhancing the function of CD8 T cells, CD4 T cells. Also it stimulates M1 shift from instead of M2. M2 is suppressor cells, M1 is stimulatory cells. This is because of autophagy. So autophagy stimulates M1 rather than M2. And immune cells like liver cells and the kidney cells also, they rely on glycolysis to sustain their clonal expansion and function. And naive CD8 T cells are different than activated T cells and the memory CD8 T cells. Actually, naive CD8 T cells are similar to memory CD8 cells in using of glycolysis and oxidative phosphorylation. So mainly they use oxidative phosphorylation. Activated T cells, upon interaction with antigen, they upregulate aerobic glycolysis and glutaminolysis. Memory CD8 T cells switch back to fatty acid oxidation. So here is mostly fatty acid, here is glucose in activated T cells. And this actually what happens in this is a glucose transporter, transport glucose, and then glucose to pyruvate, and then and the glutamine also in mitochondria is transferred to mitochondria form glutamate, and this induce the required energy in cells. This in naive T cells is immediately upregulated, and it switched into oxidative stress in case after activation of T cells and the binding of TCR to a certain antigen in presence of CD8 T cells. This is results in the switch and metabolic signature of T cells. So this rapid switch from naive in metabolism from naive to CD8 T cells require a certain mechanism, and in fact we worked on the metabolic signature of T cells in naive and the activated T cells, and we found in tumor-bearing mice, we inoculated mice with tumor, early tumor, and and then waited for tumor growth. and divided the animals and to sacrifice after 1 week, 2 weeks, 3 weeks and 4 weeks of tumor growth with or without cisplatin chemotherapy. And we looked at the enzymes required for glycolysis and gluconeogenesis, and we found that tumor itself we found, of course, glucose. I'm not going deep into the results, but overall we found that glucose is consumed rapidly by activated T cells, especially in presence of tumor, either starting from 24 hours out in T cells harvested from animals and cultured in vitro for 24 hours, or 72 hours, either from tumor, 1 week tumor, 1 week plus cisplatin, 2 weeks alone with tumor, or plus cisplatin. So it seems that the signature of T cells can be enhanced by the impact of fasting, and this is what we are going to work on in the next days, and this will be very interesting subject to work on. Fasting and stem cells. Previous studies actually showed that fasting can increase hematopoietic stem cells or mobilize stem cells from bone marrow into peripheral pool, and this is either even in aged mice. So this is very interesting. So to show the prolonged fasting and refeeding cycles through decreasing IGF, PKA, induce functional and balanced immune system through increasing hematopoietic stem cells and the lymphoid cells and the myeloid cell number as compared to the aged mice with chemotherapy. So the fasting can enhance mobilization of stem cells and their differentiation into myeloid cells and the lymphoid cells, even in aged mice, which is very interesting. And then recently, very interesting paper published in Cells showed that not only besides hematopoietic stem cells, fasting can induce intestinal stem cells through shaping or switching on and off the metabolic signature. So either in young or old animals, and this was very, very interesting, and of course this was mediated by enhancing of lipid oxidation. And this is a model by this paper published in Cells. Fasting, this is GIT, and fasting blocked PTM and the downstream signaling pathway and decreased mTOR, and this enhanced Banis T cells to increase the mobilization and division and regeneration capability of intestinal stem cells after fasting. So fasting blocks this pathway and release primed stem cells to division and regeneration. And this is a reversible process. So after refeeding, the excess of stem cells and Banis cells are restored and come back to the normal. So this is a kind of how fasting sustain integrity of GIT, especially after chemotherapy. And this show the effect of fasting even in aged animals on the stem cells and hematopoietic stem cells and lymphoid and tissue specific, and which results in white blood cells. It's not only white blood cells and hematopoietic stem cells, but also the intestinal stem cells after fasting and different cycles. The last thing is the fasting and the immunosuppressor cells. As I mentioned before, immune cells are elastic, so each type of immune cells are either type 1 or type 2. All type 1 either Th1, Th17, DC1, M1, N1 are good for tumor regression or anti-tumor response. And the other side, type 2 are induce tumor progression, not regression. So in tumor and the tumor suppressor cells include macrophages, which can be M1, M2. M2 are pro-tumor, wound healing, and anti-inflammatory, in opposite to M1, which induce anti-tumor and cytotoxic pro-inflammatory. And the type 2 or M2 T cells are called myeloid suppressor cells. Tumor cells produce TGF-beta IL-10, which of course block T cells, and regulatory T cells under this also produce the same cytokines, and these both block T cells, and the myeloid suppressor cells and similar to Treg cells also block T cells. Both regulatory myeloid derived suppressor cells and regulatory T cells are emerged in tumor microenvironment at high levels. My group actually has been focusing on MDSCC for almost 10 years now, and we found solid data that MDC are mobilized and become very suppressor in cancer patients, in different cancer patients, through different mechanisms. This cartoon shows that in tumor induce MDSCC, and the MDSCC produce different growth factors which induce tumor growth, as well as block all stimulatory cells and switch it into type 2. And of course, this induce T cell function through reducing IDO and tryptophan and glutamine by T cells, which are required. These two amino acids are required for T cell function. So this interaction between tumor growth, MDSCC, and the T cells and the other myeloid cells is very, very important, and can be determined for anti-tumor immunity. Besides, recent studies show that tumor can induce peripheral blood mononuclear cells released from bone marrow can be turned into MDSCC under ferroptosis. And the ferroptosis also by MDSCC induce blocking of T cell activation, blocking T cell proliferation, and induce tumor-associated macrophage, which is type 2, and this is, of course, enhanced tumor growth. So MDSCC play a critical role as immunosuppressor cells and the tumor and enhancing the tumor growth. I'm not going because of the time to talk more about that, but the accumulated evidence showed that the either caloric restriction or short-term fasting induce certain mechanism on tumor cells and in MDSCC itself to block the suppressor function of MDSCC and their pro-tumor function for tumor growth. Some effect like decreasing CD73 on tumor cells and decreasing HO, heme oxygenase, and decreasing, which is responsible for differentiation of M1 to M2, lower the glycolysis, and increasing the mobilization of lymphoid progenitors and derived T cells from bone marrow. So in overall short-term fasting induce or block the function of MDSC, and not only function but their trafficking into tumor microenvironment through decreasing the CD205, which is responsible for migration of MDSC into tumor microenvironment, and also induce some metabolic signature in these cells. And this figure show the decreasing of trafficking molecules and the glucose metabolism and increasing apoptosis of MDSC by short-term fasting, which resulted in several tumor models into anti-tumor immunity and cancer regression. Interestingly, when MDSC are infused back into the fasting tumor animal bearing tumor, the effect is blocked. So it's very clear that these cells are very important. So in human, we have been in cancer patient, we have been working in MDC, and we found high number of cells in overall cancer, and based on these cells, the number of these cells increased based on stage and even after chemotherapy, and also based on the tumor burden, more cells, either relative and absolute numbers, and this is the different stage in breast cancer.